Okay, so welcome back to this next video on the GQ pathway and calcium signals. We are looking at the moment at a G protein coupled receptor which was coupled to the heterotrimeric GQ protein, and so far in this story we've got to the point that we have activated this phospholipase C beta enzyme, and we're just about to see what it's actually going to do. So basically, the substrate for this enzyme is phosphatidylinositol 4,5-bisphosphate, and basically what it does, and what this enzyme does when it's been activated by the alpha-Q GTP complex, is it's going to cleave a bond in this PIP2 molecule. And the bond it's going to cleave is this bond between the glycerol molecule and the phosphate group, so the bond between the third hydroxyl group of the glycerol molecule and that phosphate group. So the overall products that you get out of this are, you get a diacyl glyceride molecule here, which is glycerol linked to two free fatty acids, or two long-chain carboxylic acids. So there's our diacyl glyceride. Okay, and the other molecule that you get over here is, let me just put those down, is inositol 1,4,5-trisphosphate, which is basically this inositol, this 6-carbon ring here, with 3 phosphate groups bound to it, because it received the phosphate group from the phospholipid, from this phosphatidyl group, because you've cleaved that phosphate group off of that third hydroxyl group of glycerol. So you've now got inositol with 3 phosphate groups on, and this is known as inositol 1,4,5-trisphosphate, but it's often more commonly abbreviated to IP3, and you might also hear people call it just inositol trisphosphate, IP3. Right. Okay, so we're not particularly interested in this video in diacyl glyceride. It has a very important role. It goes off and activates protein kinase C, but we're interested in the calcium signaling that results from the GQ pathway, and the calcium signaling results as a result of this IP3 molecule rather than as a result of the diacyl glyceride molecule. So we're going to focus our attention on inositol 1,4,5-trisphosphate. Okay, so now what we need to look at what this molecule actually does. Well, basically, if this is the endoplasmic reticulum of the cell, denoted here as a box, okay, and I'll draw it actually in my initial drawing of the cell. So basically, if we have our initial drawing of the cell, maybe this is the endoplasmic reticulum. Okay, so I'll label it up as the endoplasmic reticulum. Now, basically, the endoplasmic reticulum acts as a store of calcium. Calcium concentration is extremely low in the cytoplasm of a normal cell. In the cytoplasm of the normal cell, the calcium concentration is usually around 100 nanomolar, so very, very low calcium concentrations. And when I put those square brackets around calcium, it means concentration of calcium, and that I there denotes intracellular. Okay, so the cytosolic calcium concentration is usually around 100 nanomolar, very, very low, whereas the concentration in the ER lumen is much, much higher. So, if you want to produce a calcium signal, you can release calcium from the ER lumen, basically. And there exist channels in the membrane of the endoplasmic reticulum which allow you to do this. And one of these such channels is something known as the IP3 receptor. So here, I will draw the IP3 receptor. So basically, the IP3 receptor is made up of 4 separate subunits. Okay, so 4 separate proteins all come together to make this IP3 receptor here. Okay, and there are 3 different genes, all coding for proteins which can make up a quarter of this IP3 receptor. And these are massive great proteins that they code for. These proteins have a size of around 2,750 amino acids, basically. So they are enormous, great big proteins. You can either use one gene to encode for 4 proteins which are all identical and put them together to make a homotetramer, or you can make heterotetramers where all 4 subunits are not the same, basically. They're not encoded by the same gene. Okay, so now let's discuss what inositol 1,4,5-trisphosphate does to the IP3 receptor. Well, basically, if we look at the IP3 receptor from the top, so where should we draw this? We'll draw it over here. If we look at the IP3 receptor from the top, so that we're looking down at its 4 different subunits like this. Okay, and bear in mind this is a cartoon. It's not, the IP3 receptor does not actually look like this beautiful cylinder I've drawn here. Okay, well basically, before IP3 binds, there are calcium binding sites available on the IP3, never mind. And there are calcium binding sites, which I've shown in orange, available on these IP3 receptor subunits. However, they are what are known as inhibitory calcium binding sites. So this is an inhibitory calcium binding site. So before IP3 has bound to the IP3 receptor, there are inhibitory calcium binding sites available on the cytosolic side of the IP3 receptor. Okay. Now, if calcium binds to these, it will make the IP3 receptor more likely to be in the closed state, basically. So it will inhibit the IP3 receptor. Now, there are also IP3 binding sites on these IP3 receptor subunits, which I will denote like so. So these in pink are the IP3 binding sites. So when IP3 molecules go up in the cytoplasm as a result of this GQ pathway, IP3 will come and bind to these IP3 receptors, and one IP3 molecule will bind to each of the 4 subunits. So you'll overall have 4 IP3 molecules binding to the IP3 receptor. And when IP3 binds, what will happen is it will cause a change in the conformation of these subunits that make up the IP3 receptor. And this inhibitory calcium binding site will go away, basically. It's magicked away. And it's replaced by a stimulatory calcium binding site. So I'll denote this in green. So this in green denotes this stimulatory calcium binding site. Okay? And now, if calcium binds to this stimulatory calcium binding site, of which there are 4 separate stimulatory calcium binding sites. So you'll need a calcium to bind to all 4 stimulatory calcium binding sites. Okay, so you'll need 4 calcium ions overall. Basically, if calcium does bind to those stimulatory calcium binding sites, however, then the receptor will open and it will conduct calcium through it. So it is a common misconception among medical students that IP3 actually opens the IP3 receptor. No. What it does is it changes the conformation of the IP3 receptor so that calcium binding to the IP3 receptor now opens the IP3 receptor rather than closing it, increasing the probability that it's closed, basically. Okay, so it takes away this inhibitory calcium binding site and produces a stimulatory binding site. Okay, right. So, if calcium comes in and binds to these stimulatory binding sites, what will now happen is that this IP3 receptor will open and you will release calcium from the endoplasmic reticulum into the cytoplasm. Now, you will release a huge amount of calcium, quite a lot of calcium compared to the amount that's in the cytoplasm usually. You will release a lot of calcium from the endoplasmic reticulum through these IP3 receptors, and that causes a calcium signal. Now, the important thing to understand is that there is always some calcium ions in the cytoplasm. So it is always possible that these IP3 receptors will get some calcium binding to those stimulatory calcium binding sites and then causing it to open. Okay, so if IP3 does come along, generally what's going to happen, therefore, is that the IP3 receptors are going to open and calcium is going to come out. Okay, so let's look at the different types of calcium signaling that you can achieve then. Right, so there are lots of different overall calcium signals that you can produce from this same starting point, this GQ pathway. And basically, what's different between them is the amount of stimulation that you give the cell in the form of this GQ pathway. And we'll continue this discussion in the next video.